The cell is regulated by a molecular control system. There are genes that turn off and turn on as they talk to each other and to the cell, regulating what the cell is doing at each phase. The textbook uses a mechanical analogy of how this happens. I'm going to go with the road analogy, since you may need a different one from that presented in the book. The cell is like a car driving down the road towards cell division, and it has only one road it can go on. It has to drive over the pavement, one block at a time. It can't skip over stretches of pavement. Just like a car on a road, the cell has to travel over each event in the cell cycle. It can't skip one along the way. The next one can't happen until the one before it completes. There are checkpoints where the cell can stop and stay where it's at. These are the traffic lights that can stop our cell car. If the light stays red, the cell stays in that phase. If the light turns green, then it gets to move on down the next stretch of road to the next traffic light. Let's use some examples you may be familiar with. The cells in the dermal layer of your skin, that's down under what you see on the outside, are continually dividing. They have to replace dead skin cells we are constantly shedding. So the car cell has a very short to no red lights on each of the checkpoints, so they can keep replacing your skin. You wouldn't want to wear a hole in your skin, now would you? Your liver cells, on the other hand, don't have the kinds of physical stresses that your skin does. You aren't rubbing your liver up against elevator buttons and against clothing, so it doesn't need to replace worn away cells. Generally, your liver cells divide every year or so to replace cells damaged or worn out from working full-time with no vacation at their liver cell jobs. What happens is, when the liver cells hit this checkpoint, they have a long red light that keeps them in G1. In G1, they make enzymes the body needs. They store and release glycogen and other liver cell stuff. Some types of cells, once they mature, we thought never divided again. The traffic light in G1 turns into a parking lot. We call this G0, or eternal G1 phase. We thought nerve cells were like this. As it turns out, they divide about every 10 years. Not fantastic for those with spinal cord injuries, but if we can figure out a way to turn that red light into a green light, we might be able to repair spinal cord injuries, and para and quadriplegics will be able to be healed. But sometimes it's not a good thing for the red lights to turn green. Sometimes a mutation can turn a red light green when it shouldn't be. Or a cell may be damaged by various environmental factors such as UV radiation or toxins, and it may still live. It is damaged at the checkpoints, and it may be turned permanently on. So the cell is continually going through the cell cycle unregulated. In any case, the cell becomes cancerous with uncontrolled multiplication of cells in multicellular organisms. The mass of uncontrolled cells start pressing on cells that are still doing their jobs, squishing them to death. They also take nutrients, starving the non-cancerous cells. Either way, it's a bad situation. I'll leave you to look up the terms benign, malignant, and metastatic in the textbook. Well, that's it for this chapter. Let's go over the learning objectives for this chapter again. Describe the structure and organization of the prokaryotic genome and eukaryotic genome. List the phases of the cell cycle. Describe the sequence of events during each phase. Draw or describe the mitotic spindle, including centrosomes, kinetochore, microtubules, non-kinetomicrotubules, and asters. Compare cytokinesis in animals and plants. Describe the process of binary fission in bacteria. Explain how the abnormal cell division of cancer cells escapes normal cell cycle controls, and distinguish between benign, malignant, and metastatic tumors.